Class 11, Chapter 6, Anatomy of Flowering Plants. You can very easily see the structural similarities and variation in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to internal structure and functional organization of higher plants. Study of internal structures of plants is called anatomy. Plants have cells as the basic unit. Cells are organized into tissue, and in turn the tissue are organized into organs. Different organs in a plant show difference in their internal structures. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. The tissue system. We were discussing types of tissue based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would be dependent on location. On the basis of their structure and location, there are three types of the tissue system. These are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant and comprises epidermal cells, stomata and epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layer. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with the waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cell, which enclose stomatal pore. In the grass, the guard cells are dumbbell shaped. The outer walls of guard cells away from the stomatal pore are thin, and the inner walls towards the stomatal pore are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells and the surrounding subsidiary cells are together called stomatal apparatus. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongated elongation of the epidermal cells and helps absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays. In the primary stem and roots. In the leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. Now, the vascular tissue system. The vascular tissue system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stem, cambium is present between the phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundle. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundles are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stem and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. Now, anatomy of dicotyledonous and monocotyledonous plant. For a better understanding of tissue, organization of roots, stems and leaves, it is convenient to study the transverse section of the mature zones of these organs. Dicotyledonous root. Now in figure, it shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of the epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular space. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cell without any intercellular space. The tangential as well as radial wall of the endodermal cell have a deposition of wall impermeable waxy material sovereign in the form of Casparian strips. Next to endodermis lie a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between xylem and phloem. All tissues on the inner side of the epidermis, such as pericycle, vascular bundle, pith, constitute the stele. Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. It has the epidermis, cortex, endodermis, pericycle, vascular bundle and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. Dicotyledonous stem. The transverse section of typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide the mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called endodermis. The cells of the endodermis are rich in starch grain and the layer is referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is characteristic of dicot stem. Each vascular bundle is conjoint, open and with endarched protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Monocotyledonous stem. Monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located one. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Now, dorsoventral dicotyledonous leaf. The vertical section of dorsoventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and the lower surface abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. Now, the adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round or loosely arranged spongy parenchyma is situated below the palisade cells, which extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular systems includes vascular bundles, which can be seen in the veins and the midriff. The size of the vascular bundle are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves, and the vascular bundles are surrounded by a layer of thick wall bundle sheath cells. Look at the figure and find the position xylem in the vascular bundles. Now, isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that the dorsoventral leaf in the many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surface of the epidermis, and the mesophyll is not differentiated into palisade or spongy parenchyma. In grasses, certain adaxial epidermis cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are fleshy due to water stress, they make the leaves curl inward to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar size of vascular bundle, except in main veins, as seen in vertical section of the leaves. So that's it. Thank you so much.